Hello guys, welcome to Medification. Today we will start with renal physiology and our first topic will be PCT, that is proximal convoluted tubule. Now as all of you can see, I have drawn here a single PCT cell. PCT cell are brush bordered cell and they help in increasing the surface area for having more resorption. So it is an important point. And second point is that PCT cell have increased mitochondria. Why? To have active transport. Second, PCT is the area where the maximum in a nephron. PCT is the area where the maximum resorption will take place. And near about 70% NaCl is resorbed here, 70% of potassium is resorbed here, and for water we will see in the next slide. So now we will see how water gets resorbed in the PCT. Actually we can learn in this way that if we consider a PCT cell, we can learn this way that when sodium gets resorbed, then water also follows it. So we can also say that 70% of the water is also resorbed in the PCT, as sodium is also resorbed in the PCT. And how much is resorbed? It is 70%. And another point is that, which is the main important point, water gets resorbed by osmosis. Very important point. And in PCT, water reabsorption is not under any obligatory process. Now we will come to glucose resorption. In PCT, glucose is the only substance which is resorbed 100%. Like all of the glucose get resorbed in the PCT itself. So how does it occur? It occurs by the sodium glucose co-transporter channel, or we can say denote it as SGLT2. And this type of co-transporter channel is found only in the PCT. And this process by which the sodium and glucose get resorbed by the SGLT, it is called secondary active transport. Now before starting loop of Henle, I would like to say 2 points, which is 70% of the sodium and 70% of the water which is resorbed in the PCT, and the rest which passes on is the isotonic solution. But in the loop of Henle, the main concentration and dilution of urine occurs. Now we will study an important topic, that is countercurrent mechanism or system, as you say it. Occurs not only in the loop of Henle but also in the limbs, testes and intestines also. So as all of you can see here, this is the descending loop of Henle, and first initially at the starting the total concentration of urine is 300 milliosmol. So now at the initial level it is 300 milliosmol. So after that we have to note a point that descending loop of Henle is permeable to water, but it is impermeable to sodium. So what will happen next? Water will move out. You can see here the aquaporin. I know the drawing is terrible, but still. So water moves out, and then so as water moves out, what will happen? The sodium concentration will be increased, or rather, I would say that there will be urine concentration occurring. So first it was 300 milliosmol, then it becomes 600 milliosmol, and as it gradually descends, it becomes 900 milliosmol, and by the time it reaches the inner medulla, it becomes 1200 milliosmol. So by this we can also say, and we can also see here, that descending loop of Henle is the concentrating segment. And a point we should note down here is that in the inner medulla, it is a point, it can be referred as counter current multiplier also, because you can see that the concentration is increasing from the initial level till it is gradually descending to 1200 milliosmol. So we can say that descending loop of Henle is the concentrating segment. Now after it reaches here, and one point to be noted here is descending loop of Henle is thin and ascending loop of Henle is thick. So now we will see that through a channel which we also call a Bartin, the channel name I am saying. So sodium, potassium, and 2-chloride ion can go out. So here the opposite of what was happening in the descending loop of Henle is occurring, that is dilution is occurring. So we can say that ascending loop of Henle is the dilution segment, whereas the descending loop of Henle is the thin concentrating segment. Here also in the ascending loop of Henle, water is impermeable, but it is permeable to sodium. So gradually as more number of solutes are moving out, so dilution is occurring, and so you can again see that gradually from 1200 to 900, 600 to 300, concentration is decreasing. So we can also say that, as I said earlier, that loop of Henle is the point where concentration and dilution of urine take place. Another point I would like to say is that if I'm drawing over here a descending loop of Henle, this thing rather, and over here all of us know that it is the Vasa recta. Now, important point to be seen here is that if here the concentration is 300 milliosmol in the descending loop of Henle, then in the Vasa recta, here also the same concentration will be there. So, if here 300, here 600, then here also 600. But what is the significance? What is the significance of Vasa recta which is occurring here? It is that Vasa recta prevent washout. It prevent the solute washout from the descending loop of Henle. So, Vasa recta has the same level of osmolarity as the descending loop of Henle. We'll start with Raas or Renin angiotensin aldosterone system. It is a very important topic, but first we have to see what is JG apparatus. JG apparatus is nothing but it generally consists of JG cells, macula densa, and extraglomerular mesangial cell. So what is the function of Raas is that Raas gets activated due to mainly two reasons: when there is hyponatremia, hypotension, and sympathetic nervous system activation. So 3 points, what are they? Hyponatremia, hypotension, and SNS activation. So what happens is that first we will see that is the function of JG cells over here, like taking like this is the afferent arteriole, and like JG cells are present like this. So we are not taking all the cell. I'm drawing here a single JG cell, and here actually inside the JG cell granules will be present, and within the granules renin is stored. So we can say that another name for JG cells is also granular cell. My handwriting is terrible here. Anyways, so the JG cells have the granules which releases renin. Now, what is the function of renin? First, when I said, as I said previously, like in the cases of hyponatremia, hypotension, or due to SNS activation, our RAS system gets stimulated. So, the angiotensinogen which comes from the liver When it comes near the kidney, renin acts on it and it gets angiotensin 1. But most important point is that it is an inactivated form. So, after angiotensin is formed, angiotensin 1, angiotensin converting enzyme from the lungs act on it and convert it into angiotensin 2, which is a active form. And so angiotensin 2 is a vasoconstrictor. So what will happen as a result? Blood pressure will increase. And another thing which angiotensin 2 does, it acts on the adrenaline gland and stimulate it to release aldosterone, which in turn increases sodium resorption. So this is the main overview of the Raas system. So now let's see some important question. So the first question, countercurrent mechanism in the kidney is responsible for. The first option is absorption of glucose. No, it won't be the answer because we have discussed that 100% of glucose reabsorption takes place in the PCT. Maintenance of blood flow will not be the answer. Yes, C will be the answer. Osmotic gradient in medulla. Why we will discuss, like we discussed earlier, in the descending loop of Henle, concentration of sodium takes place as water is permeable but sodium is impermeable. So descending loop of Henle is the part where concentration of the urine takes place. So the answer will be osmotic gradient in medulla. Juxtaglomerular apparatus lies in relation to. So juxtaglomerular apparatus are collection of specialized cells which lie in close relation to glomerulus. Yes. So the third question is, hyperosmolarity of renal medulla is due to, obviously it's sodium. Why sodium? Because we have seen that in descending loop of Henle the main concentration of the urine takes place, and as in the descending loop of Henle water is permeable and sodium is impermeable, so hyperosmolarity occurs in the renal medulla due to sodium. Initially in the descending loop of Henle the concentration is 300 milliosmol, and in as it descends gradually the concentration increases to 1200 milliosmol. So that's all for today. Thank you guys.